Hi everybody. Today we are going to talk about the type and structures of RNA, and among them we have already discussed the mRNA and the tRNA. Now let's go to the next one, the ribosomal RNA. Ribosomal RNA is, as the term implies, is a rRNA which has been found associated with the case of ribosome. Now, in the case of prokaryotes, the ribosome has a different subunit, as well as in the eukaryotes, they have a different subunit. Now, prokaryotic ribosome, it mainly consists of two subunits, that is a small subunit called the 30S subunit, as well as a large subunit called the 50S subunit, which will get assembled to form the 70S subunit, 70S whole unit. Okay, so in between these, what happens? There are certain certain rRNAs attached, and that's what we are going to talk about now. Now, there are mainly you have 23S ribosomal rRNA and the 5S ribosomal rRNA, which is being associated with a 50S subunit. And to the small subunit, that is a 30S subunit, you have the 16S ribosomal RNA and 21. So coming to the general features of ribosomal RNA, like all kinds of RNA, ribosomal RNA is also being transcribed from a template strand of DNA. And about 0.1 to 0.2% of the bacterial genome is estimated to be responsible for the transcription of this ribosomal RNA. And we can find that some well-conserved regions are usually being found in this, which contains methylated residues. Now, in the case of 16S ribosomal RNA, we can see that 10 methyl groups are being present in it towards the 3' terminus, and approximately 20 methylated residues are being found in the case of 23S ribosomal RNA. Generally, the ribosomal RNA will participate in the protein synthesis, but it will not code for any of the proteins. It mainly assists the ribosome in the protein synthesis. Now, how it assists, that's what we are going to see. The specific sequences in the ribosomal RNA will help in the initial attachment of the mRNA to the 30S subunit and assure the process of translation. And the sequence in the 3' terminus of the rRNA interacts directly with the mRNA at initiation. Now, if you go to look at the 16S rRNA, it has been found that specific regions of this 16S rRNA will directly interact with the anticodon regions of the tRNA in both the A site and the P site. A site and P site, that is in the transfer RNA, there are certain regions called the A site and the P sites. Okay, so in that region of the tRNA, that is in the anticodon region, there are certain specific sequences. That specific sequences are being interacting with the 16S rRNA specific regions. And if you go to consider the 23S rRNA regions, that interacts with the CCA terminus of the peptidyl tRNA. So, the tRNA is the one which is bringing the amino acid towards the ribosome. And within the ribosome, you have all these rRNA regions, such as the 16S rRNA as well as the 23S rRNA. So, the topmost region, that is at the 23S rRNA, that is interacting with the CCA terminus of the tRNA, and at the bottom of the tRNA we have the anticodon region, and that anticodon region is interacting with the 16S rRNA, which has been found attached to at the base of the ribosome. Now, coming to the mammalian rRNA, mammalian ribosomal RNA, as a ribosome, it also has different subunits. The mammalian ribosome has two different subunits, like 60S subunit and the 40S subunit. And here you can find that attached to the 60S subunit, there is the 28S ribosomal RNA, the 5' ribosomal RNA, and a 5S ribosomal RNA. and attached to the 40S subunit is a 18S ribosomal RNA. And also various proteins are also being associated with them. Now, the mammalian ribosome contains two major nuclear protein subunits. As we told earlier, it has having a larger subunit and a smaller subunit. That's already I have told you. And the ribosomal RNAs which have been associated to that also be mentioned. Okay, so the 16S subunit has what? 5S ribosomal RNA, 5.8S ribosomal RNA, and 28S ribosomal RNA. Okay, this is all mentioned earlier. And the function of the ribosomal RNA molecule in the ribosomal particle is not fully understood here, but they are necessary for the ribosomal assembly, and they play a key role in the binding of the mRNA to the ribosomes and its translation. And recent studies, they also show that some of the rRNA component, they have a peptidyl transferase activity, and thus it is an enzyme called the ribosome. Now we are going to deal with the small RNAs. Now, most of these molecules are complex. These are mainly found in eukaryotes, but some of them can also be found in certain bacteria and bacteriophages and all. So, these molecules are complexed with proteins to form the ribonucleoproteins and are distributed in the cytoplasm as well as in the nucleus or in the both. And they range from a size to about 20 to 300 nucleotides, and they are present in 100,000, that is units, tens, hundreds, thousand, 10,000, lakh, yeah. 1 lakh to how many cap? Just look, units, tens, hundreds, thousand, 10,000, lakh, 10 lakh. 10 lakh copies to 1 lakh to 10 lakh copies per cell. So coming to the small RNA. Small RNAs, they are of different types. Okay, they can be of SNRNA, then next one they can be of microRNA, then they can be also of about saRNA. So now let's start with the first one called the SNRNA. SNRNA is mainly involved in the mRNA processing and gene regulation. And you can see as a picture is being found that here there is exon, intron and exons. So you need to remove these introns. And these introns are mainly removed with the help of SNRNAs. Okay. And here this SNRNAs U1, U2, U4, U5 and U6 are mainly involved in the intron removal. And the processing of the hnRNP into the mRNA also being done with the help of these SNRNAs. And the U7 SNRNA is mainly involved in the production of the correct 3' ends of the histone mRNA, which lacks a poly A tail. So look at this picture. You can find that exon, intron and exon. So where does this happen? This mainly happens in the case of eukaryotic forms. So SNRNAs, they mainly help in the removal of the introns as well as also at the correcting of the 3' end, the histone mRNA, that is which lacks the poly A tail. This U7 SNRNA is involved in the production of the correct 3' end of the histone mRNA. Now comes to the next small RNA called the microRNA and the SIRNA. Now microRNA is this you can find in certain, it is found in almost all the living forms, and here they are short non-coding RNAs, and they have been found to regulate the expression of certain proteins. Here what happens, once the microRNA goes and binds onto the mRNA, what happens, the translation of that particular protein will not happen. So the blocking of the translation process can be done by the help of these microRNA regulators. Now coming to the SIRNA. These are called the small interfering RNAs, and they are 20 to 25 nucleotide long double-stranded RNA molecules that have a variety of roles. Now some of them are involved in the RNA interference pathways, where it interferes with the expression of a specific gene. by hybridizing to its corresponding RNA sequence in the target mRNA. This then activates the degrading mRNA. Once the target mRNA is degraded, the mRNA cannot be translated into the protein. So what happens? Once here, the siRNA, okay, that will go and bind. It's a silencing RNA, and once it is being bound, what happens? Dicer also comes and bind, and the cleavage, the breakdown of that part. Once the siRNA is being bound, what happens? A dicer, that is a RNA cutting enzyme, will also be bound, and then the mRNA breakdown will be happened. So they are short. They are usually short perfect RNA. They form short RNA-RNA hybrids with their distinct targets potentially anywhere within the length of the mRNA where complementary sequence exists. And the formation of such rRNA duplexes between the siRNA and the mRNA results in reduced specific protein production because the siRNA and the mRNA complexes are degraded by the dedicated nucleolytic machinery. And some or all of this mRNA degradation occurs in specific organelles called the P bodies. So these are mainly found in the eukaryotes. So when the siRNA goes and binds to the complementary, it's like it won't be able to bind at all regions where it has been complementary, where it finds a complementary strand, there this on the mRNA, this siRNA will go and bind. And once it binds, what happens? Certain various other nucleotide breaking enzymes such as the dicers come and bind onto it, and the mRNA SIR onto this miRNA and SIR RNA and mRNA complexes. Onto that the dicer comes and binds and it degrades it, and so then the mRNA translation will not be happened here. So this miRNA as well as the siRNA, they are being used as potential targets for therapeutic drug development in humans, and they are also frequently used to decrease or to knock down specific protein levels in experimental procedures in the laboratories as an extremely useful and a powerful alternative to gene knockout technology. So we need to conclude what are the functions of RNA. RNA is a nucleic acid messenger between DNA and ribosomes. It serves as a genetic material in some organisms such as viruses. And some RNA molecules play an active role within the cell by catalyzing biological reactions, controlling the gene expression, and sensing and communicating the responses to the cellular signals. And the messenger RNA, it copies the DNA in the nucleus and carries the information to the ribosome. And the ribosomal RNA makes up a large part of the ribosome and reads and decodes the mRNA. The transfer RNA carries the amino acids to the ribosome where they are joined to form the proteins. And certain RNAs are able to catalyze chemical reactions such as cutting and ligating other RNA molecules, and the catalysis of peptide bond formation in the ribosome could also be carried out by these, and these are known as the ribosomes. I hope this session is understanding for you. If you have any queries, you can contact me. Thank you.